Larry, you're on. Larry, you're on.
Larry went
Marianne, can you call the roll, please, before we get
Yes.
going?
Got it. Yeah, you were on mute for one minute. Well, I'll go ahead and call the roll now. Maria Bonneville,
Present.
Leandra Clark Harvey, Mark Fisher Colbury, Fred Fisher,
Fred is here.
Judy Gasson,
Here.
Larry Goldstein,
Here.
David Higgins, Vito Bassiani.
Yes, Veni Vidi Vito.
All right, Steve Julesgaard. Steve? It's not there.
He is.
Okay.
Yeah.
Present,
Okay,
sorry.
gotcha. Okay, Pat Levitt,
Present.
Lauren Miller Rogan, Marvin Southern, Keith Yamamoto.
I'm here. Hi, Rusty.
Hey.
All right, we're good, and we do have a quorum, Larry, so you may proceed.
Great, thank you. So 2 items today. The first will be a talk from Rusty Gage that I'll introduce in a moment. And the second is we'll hear a concept plan from Rosa Canet-Avalos laying out a possible funding system for neuropsychiatric stem cell research. So Rusty will speak for about 45 minutes, including questions, and then we'll get Rosa up. So Rusty Gage, Dr. Fred Gage has been a longtime colleague of mine. He has a number of firsts over the years, including evidence that there's neurogenesis in humans, some wonderful stem cell work over the years. And the reason I thought we should hear from him today is as we launch into neuropsychiatric stem cell research, it's important that we be able to analyze what goes wrong in mutations that predispose to schizophrenia or bipolar disorder or any of the diseases that we may turn out to be interested in. And Rusty's lab recently published what I think is one of the most advanced phenotypic systems for evaluating what is happening inside of brain organoids. And with no additional verbiage from me, I'm going to turn it over to Rusty. So take it away.
All right, I'm screen sharing now. Let me see if you can see that. Okay. Well, thanks for inviting me to present. I'll be presenting our efforts to develop a fully human brain organoid model system. It'll be mostly methodological, but there are some conceptual things that I'd like to advance as well. So here's the layout. What are our goals? Well, there's a lot of focus lately on the last 10 years or so on developing human brain organoids, in part because of the, let's say, lack of complete satisfactory evidence from mouse models, especially of psychiatric diseases, and the ability to look at human brain tissue and models of human brain is hoped to advance our ability to make new discoveries. So with that recipe for the argument for the organoid, human organoid, there's a couple of things that are problematic so far, and that is that the organoids that have been and are being used in vitro are not vascularized. And what happens because of the 1 millimeter rule, that means things can't diffuse more than 1 millimeter into the organoid. Once they get larger than 2 millimeters in size, the core begins to rot. And as the larger they get, the larger the core becomes. And in my mind, in our mind, our lab, it's very hard to make conclusions about the outer rim of an organoid when you know that the inner core is dying. We need to vascularize the organoids, eliminate the necrotic core. Next is most of the organoids that currently exist are really neural organoids. They're made up of neurons or neural precursors and are not really imbued naturally with other cell types. And of course, the brain is made up of many different cell types. And And once you have these, as you imbue the organoids with these different cell types, you need to establish their health and function and demonstrate that they are responding as one might expect them to respond in situ. I'll give you evidence today on neurons, microglia, and astrocytes, and in progress for endothelial cells and pericytes, which are the cells of the blood-brain barrier or the vasculature. And then with a long-term plan of myelinating the axons within the organoids. Now, the goal, obviously, is as we move through this, we can begin to compare healthy brain to disease brain. And really, it's disease agnostic, because with IPS technology, you can get fibroblasts from any patient available. Once there, you can do what we call inductive experiments, where you can replace a healthy cell with a disease cell to determine if that disease cell is driving or a consequence of, or activated as a consequence of the environment that they find themselves. And I'll give you an example of microglia and autism. Restoration is another approach, and that is where you have a diseased brain organoid and you replace with healthy cells to see if you can eliminate some of the pathology that exists within the organoid diseased brain. And of course, with this system, because it's living and modular, we can begin to identify cellular and molecular mechanisms that are causing or preventing these cells to become unhealthy. And in the final case, we think of this model system as a tool to test therapies in the human brain and perhaps of the patient that is being treated. So taking a fibroblast from the patient that's going to have some sort of therapeutic intervention, you can actually test the drug, test the vector, test the gene therapy, test the cell therapy in the organoid prior to going into the patient. So with that, we can say that there are many different cells of the brain. Most of the organoids that exist have these. Can you see my little arrow there?
Yes.
Okay. We do see radial glia in the existing organoids, and they give rise to neurons. Very late in the process, usually 4 or 5 months into it, you can see astrocytes forming in the outer core, but they haven't been tested yet for their functionality. And no OPCs or oligos have been detected so far. I'm going to start by talking to you a little bit about microglia. These are cells that are not even derived in the brain, but they're derived in what's called the yolk sac, very early stage of development, where they migrate into the primordial brain, take up residence and become, they proliferate actually when they're there, and they take up residence. Once the parasites form on the outer edges of the endothelial cells and prevent the growth of these cells or the movement of these cells into the brain, you rely pretty much on the existing microglia that came in early on. However, bone marrow-derived macrophages or myeloid-erythroid cells can migrate into the brain also under certain circumstances. So we have two sources, early development and late development for microglia. But again, they're not brain-derived, they are generated from outside the brain. A lot of interest in microglia lately suggested their role in a variety of diseases, particularly their role in inflammation. And so a lot of work has been put in trying to characterize them. And of course, fabulous work by Ben Barris early on, establishing what is a microglial identity, both developmental and mature. We were involved in isolating microglia directly out of the brain, but one of the things that happens with microglia in vitro is they're very sensitive to the environment. And when they go into an in vitro setting, they downregulate key proteins that are thought to be essential for the features and function of microglia, including these genes here. I'm not going to go too much into specific genes unless there's interest from the group. I can give you more detail in the discussion section, but the fact is they are microglia in vitro, but they're not fully mature. So what we've done is we've taken our regular organoid and we make erythromyeloid cells. So we take a fibroblast and turn it into an iPS cell, and we generate basically early development hemopoietic cells, and then drive them slightly into the phase where we know from human development that they migrate into the brain at about 3 to 4 months, weeks of age. And so here we have the green as the organoid, and through we have our induced myeloid progenitor cells on the outside, and you can see in the movie that they're migrating in over a period of time, and they settle in the brain. Now, we did, I'm not going to go through it, but we and others have done a series of experiments trying to get microglia into the organoids, and they don't survive very well. They require several human-derived proteins that protect the survival of the microglia, and without them, they don't survive. And even with those added proteins in vitro, the microglia do not survive more than a month, and they are in what's called a reactive state, so they're not naturally formed. We found we developed a model in 2018 where we can take an organoid and we can form it and then transplant it into the retrosplenial cortex, overlying a vascular bed. And so it's overlying the superior colliculus, and the blood vessels from the superior colliculus go in to the brain, to the human organoid. And you can see here, after interorbital injection of dye, that the blood-brain barrier is formed, and there's no leakiness into the host. When we transplant the microglia enriched organoids into the brain, we see lively, fully mature microglia. These are labeled with TD tomato here, but double labeled with IVA1, a marker for microglia. We can do this in some detail quantitatively. And now we can see in vivo that these genes that previously were not on in vitro are now being expressed in the microglia, so in vivo setting. Interestingly, the CF1 receptor, which is a survival receptor and gene for microglia in humans uniquely to mouse, is being expressed in our organoid, human organoid, so we do not have to add or engineer the cells in any way. They are naturally surviving. IVA1, here are these markers for the additional markers for microglia. We do, to sort of confirm that they really are in a mature form, we have looked at single cell sequencing at 6 weeks, 11, and 24 weeks after transplantation. And we do our UMAP analysis here, and you can see that these are color-coded for the sensome genome. So Hickman some time ago extracted human microglia from the human brain and analyzed genes that are important for what is called a sensome. So microglia have this interesting property where they sense danger or damage in the environment, and they attack, destroy cells that may be detrimental to the cell. What is conceptually built into this is that when microglia become overactive, they begin harming or hurting the existing healthy brain as well. So we're seeing that there's an upregulation of these sensome genes. They become more active, and also genes that we and others have discovered to be important for maintaining the environmental integrity of the microglia. So one of the things that we can do with this model is we can put a glass cover slip over the top of the mouse's brain. These are non-skin mice, so they're immunocompromised. And we put a 2-photon microscope over the top of the anesthetized animals, and we can watch the microglia as they sense the environment. This was an experiment done 10 years ago in mice for the first time by Axel Nimmerham, and he's now a faculty member at the Salk, and was able to see human microglia behaving in many ways the same way. What's interesting, they survey their environment, but they don't touch each other, but it's sensing for toxins in the environment. We know these cells are responsive, so we can give an inflammatory injection of what's called lipopolysaccharide, which induces an inflammatory response. And here's the control, and here's after 24 hours after injection. And they round up and appear much more like a phagocytic cell. These phagocytic cells are what are called reactive microglia, and they're not doing their healthy job. They're actually causing toxicity, secreting a lot of unhealthy molecules into the environment. Now, interestingly, when we kill a cell right between two of these, or four of these microglia, we end up seeing how the microglia actually respond. So normally what would happen is that the mouse microglia, as we've learned in the past, migrate in and surround and destroy the cell. But human cells behave differently. They work coordinately, responding to their environment, and send out their process of retaining their position in their quadrant. So this was actually a new discovery of how human microglia behave differently than do mice. In the only two species in which we've been able to see living images of how they function. Now, one of the advantages of this that we knew we could exploit is to determine whether it is the cells themselves, let's say the microglia, which are inducing a reactivity within the host, or is the host activating the microglia to become an inflamed cell type. We've been previously working with an autistic subgroup of individuals that have large brains. These are macrocephalia. And one of the things that's been shown both in PET and in histology in these patients is they have an increase in microglia morphological responses, suggesting an inflamed state or a macrophagic state. So we made organoids from our control, and we see and imbued them with healthy these are isogenic microglia, and they behave well, they look good. However, when we imbued them with when we took the ASD, that's the autism spectrum disorder brain organoids, and imbued them with their own microglia, they were inflamed. So that's the baseline, and then the question becomes, is it the brain that's activating the microglia, or the microglia activating the brain? In this one experiment, we just did the latter piece, which is to take an ASD organoid and implanted them with neurotypical microglia versus, of course, control organoid and neurotypical organoids. And here is the control with neurotypical organoid, healthy long branches, and in this case, you see that they are activated. We can now sort these microglia and the surrounding brain area, and are beginning to understand the molecular signals from the brain which are activating these microglia. So what I've showed you so far is that microglia progenitor cells efficiently populates the developing human brain organoid. The long-term differentiation was always hampered in vitro, thus limiting their use. We developed a method for chimeric transplantation paradigm, allowing us to study human microglia alongside our human brain. They survive, mature, they acquire in vivo-like resting properties, but can respond to stimuli in predicted directions. And we can learn new things about how humans are different than adults. And we can begin to take these studies into a disease context as well. I would like to step back and say we're still missing many other cells in here. I'm showing right now some unpublished results on astrocytes. We developed a protocol where we can use a series of, we can incubate our organoids in glial promoting factors, which can induce a rapid development and maturation of glia in the organoids themselves. A gene that has been identified very early on by Lawrence Studer and his team was NF1A, is thought to be an important transcription factor that enables astrocyte differentiation. And we found that with our conditioned media, we get NF1A expression as early as 21 days in vitro. This is all in vitro. By 60 days, we have a fully represented astrocyte population throughout the organoid. And when we do single cell sequencing at 10-week-old enriched organoids, as again is in vitro, we see of course, we see excitatory neurons, inhibitory neurons, precursor astrocytes, and mature astrocytes. And I'll go into that in a little bit more detail. But from the functional point of view, you can see that they continue to mature from 3 months to 5 months. And these are just all the morphological evidence supporting the fact that they continue to grow and elaborate their processes. They are functional to the extent that they can take up glutamate, a transmitter that is normally secreted by neurons, but in the absence of this uptake, glutamate can be toxic to neurons and may, in our estimate, be part of the reason why neural organoids themselves have this certain toxicity that occurs over time, because they're not able to take away the excess glutamate. They also can elicit calcium spikes with stimulation of glutamate as well in vitro. This is all in vitro. So we transplanted these cells in our transplantation model. Here's the transplant here. And they survive very well, and they co-localize with so with this human antibody, so we know they're human. Quite remarkably, this is the in vitro organoid, and it has a greater content of astrocytes, but they're really disorganized. They're not patterned and distributed like they are in vivo and like we see in the brain. So there's actually a decrease in vitro, but they begin to pattern themselves. When you look at the difference between human and mouse astrocytes, it's really quite extraordinary. Not only are they larger, but their shape is really quite different. They have these long extended processes that are directly attracted, whereas the mouse astrocytes form this spherical shape. The long processes that extend from the astrocyte are often associated with blood vessels, and that in vivo, that's what one sees, and also what one sees in vivo. After transplantation now, we can identify not just that there's astrocytes there, but the 4 principal types of astrocytes that have been described in human literature can be recapitulated in these organoids. Specifically, upper in this, it's called the peel layer. We have these intralaminar astrocytes in their processes down. The classic protoplasmic astrocyte in the core of the tissue, and the fibrous astrocyte, which sits on the glial side, moving up into the tissue. Interestingly, there's an astrocyte which is unique to humans called the varicose projection astrocyte, and we see that one as well. That's this cell right here. It has a single bushy protoplasmic-like shape, but sends on a process that is beaded. And this is a unique feature is a unique astrocyte to the human that doesn't exist in mouse or lower species. But all the other types exist in there, and they are laminated. Interesting for those that have worked with organoids in the past, once they're transplanted and vascularized, they no longer have a rosette in the core, but rather become a full layered cortical tissue. So one of the features of astrocytes that we know is, as I pointed to you before, they send their processes and express a gene called aquaporin along the vascular bed. So here is a blood vessel here, stained for aquaporin A, which is a protein from the astrocytes. Here they are laminated. In vitro, obviously there's no blood vessels, and it's just sort of distributed diffusely around in the astrocyte. So they still make it, but it's not located to the vascular system. Here is a 3D, what this series, this image is going to show you is what's called the vascular unit, which is comprised of endothelial cells, parasites, astrocytes, and endothelial cells, and extracellular matrix, and tight junction. And this is the unit that keeps the outside blood from coming into the brain, or the blood is called blood-brain barrier. So here's a three-dimensional reconstruction of that. So these are EM sections that are sectioned at very thin and then stacked. And then we can go back and label them for you. They don't come this way. We have to color them. But it not only has parasites, basement membranes, tight junctions, and endothelial cells. Unique again to the human is the fact that the astrocyte abuts the unit and completely surrounds it by one astrocyte. In the mouse, they generally send portions of their processes and would have, say, an astrocyte here, an astrocyte portion here, and maybe a third astrocyte there. So this is a unique form of the vascular unit in human relative to the mouse. Now, is the blood-brain barrier intact? We do this by giving dextran beads to the interorbital, inject them intraorbitally, and you can see that the dye will, in the liver and the muscle, it gets out and spreads out into all the tissues. However, in the brain, you get no leakage or little leakage, and only stays within the blood vessels. So here's in our adjacent mouse cortex after the same experiment, and here's within the transplant. So we see that the biotin tracer is excluded from the tissue and is maintained in there. Evidence supporting the fact that this is a relatively intact blood-brain barrier. A caveat here is that the endothelial cells and the parasites that make up this blood-brain barrier are derived from the mouse. They're migrating in from the mouse, and we're not supplying at this point those cells. So this is a limitation.
So Rusty, we're at about 25 minutes, just to give you a more.
Perfect. I'm 2 minutes away, or
Good.
3. We have in the single cell sequencing, we can look at these astrocytes uniquely, and we find that the Go categories for the genes identify those things that we believe to be important features, like blood-brain barrier, permeability, positive regulation, vascular permeability. All these genes are upregulated in the astrocytes in these embedded transplanted organoids. Just a little more information about the maturation of them. We compared this to Ben Barris's early work where he isolated astrocytes from human, fetal, and adult brain. And we see that the in vitro has few of these more adult gene markers, more of the fetal, whereas in our transplanted organs, they mature quite dramatically over time. Story I'm not going to tell too much, but this is also part of the story, is that the astrocytes embedded into these neuronal organoids induce a rather dramatic maturation of the neurons as well. So here's the in vitro organoids, and here are neural gene ontology signals indicating these neurons, purified neurons, have now taken on a mature set. And we can see this by looking at an electron microscope where you can now see intact synapses. So this is a presynaptic terminal here with vesicles and postsynaptic density and postsynaptic terminal, which are evident in these cells. Finally, we want to test functionality. So we stimulated the animals with TNF-alpha, an inflammatory induction mechanism. We saw an increase in the subset of astrocytes and the types of genes and number of cells that took place. We look at those in quite detail, and we see that there's an upregulation of two hallmarks of inflammation, interferon gamma and TNF-alpha signaling, or NF-kappa B as well. And interestingly, a downregulation of oxidative phosphorylation, so the cells become more glycolytic in their inflamed state, which is characteristic of the literature. Fun note is we can take this same organoid and test things in more detail in vitro. So we take the astrocyte organoid, treat for TNF-alpha shortly, and look for genes that are involved. And what we find is that CD38, a key regulator of NAD+ levels in the brain, involved in inflammation and energy metabolism, is dramatically upregulated in the brain, in the subset of astrocytes. There are cancer drugs out there that specifically inhibit CD38. We can apply those in this in vitro model, and we can dramatically return the NAD plus to NADH ratio in these cells after reduction. And this also is shown by looking at the fragmentation of mitochondria, again, showing that the reestablishment of metabolic activity within these cells. So we have a rapidly derived functional astrocyte, blood-brain barrier, and functionality of the glial cells. One last thing, couple slides just to show the direction we're going. I told you that the endothelial cells were not of human origin. We have applied this protocol, which is called a generation of blood vessel organoids from human pluripotent cells by Joseph Penninger. And we applied this, and we can now sort out human parasites and endothelial cells from them. It's quite remarkable. This is what these endothelial organoids look like. And they have PDGF, they have all the markers for the cells that we want. We've developed a protocol now where we can embed the endothelial organoid with our existing astrocyte and microglial organoid, and we can now for the first time see human vascularization beginning in these human organoids. And we've just begun to transplant these cells today so that we can see if the human endothelial cell and parasites will anastomose with the mouse vasculature so we can get a more complete human blood-brain barrier. This is the summary that I started off with. We have astrocytes, microglia, and astrocytes. We're on the way to endothelial and parasites. We have a strategy for oligodendrocytes that I'm happy to share. And we've begun doing this, the comparison experiments by induction with putting the healthy microglia into a disease environment to ask directionality. And we're looking forward to developing these models that will be useful for addressing human brain diseases heretofore untapped. So with that, I want to thank two extraordinary postdocs in the lab, one for the astrocytes and one for the microglia, and they team up to work on putting these together now into a common system. I'll stop there, Larry, and escape from this. And should I stop sharing? Unless you want to look at pictures again.
Well, actually, yeah, why don't you go ahead and keep control, because it could be that a question will elicit the need for a particular slide.
Okay.
So that's terrific, Rusty. This system already looks like it's going to be incredibly valuable compared to previous 2D and neural only systems. One quick question, as these organoids become better vascularized, what do you think is going to limit their size when transplanted into the mouse brain?
Yeah, so we've been doing this now for about 5 years, and they stop dividing, so you don't have, they mature enough, because they're maturing so much, they don't have any dividing cells. We pulse with BRDU, and you'll see an occasional dividing cell, but I think it's a replacement rather than any other cell. They really are restrained by the cavity size. I think that's the same question you would ask of yourself why your brain isn't continually bursting out of your skull, is that it reaches a limit, a restrictive limit. They are about, I would say, 3 millimeters when they are grafted, and they get up to about 3.5 to 4 millimeters, and then they stop at that point.
Pretty reasonable size. Pat.
Well, that was great, Rusty. Amazing technology. So I've two related questions. One is the amount of work, the amount that you did in these experiments that you described is ginormous. It's like beyond enormous, right? It's ginormous. So from the perspective of trying to understand both mechanism, and also people have written, there's been a lot of writing about the use of organoids and stem cells for targeting and drug screening. what is your view on that in terms, because what you've described is certainly time consuming, not without its, benefits. And then the other thing that's related to that is like in your biological psychiatry paper that you wrote with Paula, or that, you did an introduction to the special issue. One of the things that Paula has emphasized is the heterogeneity, which I think is biological, right? And so how are you thinking about that in terms of the numbers that you'd need to get to a point where you feel the screenings would actually be valid and valuable?
Yeah, I think,
Two very easy questions for you.
yeah. Well, for one, we do in a setting, in one day, we can do 25 to 30 animals. So we can get good sized groups that way. We usually do subject wise, we'll do them a duplicate or triplicate. These are all graphs. This is all the graphing. At that point, unlike with tissue culture, you don't feed your cells, you just put them in the cage and they feed themselves.
Sure.
So I do think that this is a more complicated model, and what I was trying to show here with the in vitro evidence. So we injected TNF-alpha, got an inflammatory response, and we could then pursue that in the subpopulation in vitro for the short-term period that we wanted. So I think we're not saying that you want to throw out all in vitro work, but rather you can get a more authentic representation of the interactions between cells in the in vivo setting. And just like we have done in the past with mice, where you do some of the work in mice, but then once you actually get towards mechanism, then you would like to make it into a simpler, more in vitro system. So you can do mechanistic kinds of things or high throughput screening. This is not where you would do high throughput screening. This is where you would discover mechanisms and then come back later and make sure that the compounds that you found that are doing their thing do it in this context as well. So I don't want to represent that I'm promoting this over and above, but as an added tool to the arsenal.
Okay. And what about the heterogeneity issue, which
yeah, I guess I'm there is individual heterogeneity, and we embrace heterogeneity as individual differences. And we, of course, are interested in that with regards to mobile elements and ways in which diversity can be generated within tools. We have found that between individuals, so same organoids transplanted in two separate mice, but from the same patient, are similar to each other. So they cluster by subject rather than just random variation. So it's not random variation. I believe, as you said, it's individual differences, and we embrace those differences. And in our, for example, in this autism study that we've done with the macrocephalia, they are clearly different. I mean, the organoids grow bigger, they grow faster, and they have more proliferating cells in them, reliably above the age-match control. So while working with humans always adds extra variance into the setting, I believe the more you can constrain the context in which you're growing them, and also that you're providing appropriate nutrients, you can reduce that amount of heterogeneity. To be honest with you, I think that part of the heterogeneity is this necrosis that occurs in the core of the organoid that leads to variable responses in the host. And each organoid is going to be slightly different depending upon how much, necrosis there really is. So I think some others have also come to recognize this, and we're seeing that this, increase in stressful response in the core is impacting the rest of those cells. So I think variability, I don't want to say all of it, but I would say some of it comes from the the other thing, just to play on that, you can get astrocytes, human astrocytes in these organoids, but it takes about 5 months. Then they're only in the outer portions. And that's a variable point because they're not always there. So I would argue that, you know, like all of us, we want to have as much control over our environment. That's why we want to drive the astrocytes at a particular time. We want to embed the microglia at a particular time. We want to bring the endothelial cells and try to mimic the time period when they actually would get in there as best we can. A lot of words, a lot of words.
That was great. Thank you.
Questions from the group. Rusty, the progress you guys have made, I'm sure it's been agonizing at times, but certainly from the outside, oh, it looks easy, and you know, it was really fast, and you knew all along exactly what you're going to do. But it's a beautiful system, and you know, I think all of us look forward to further developments for figuring out what's going wrong and all the various disorders we're trying to fight, ALS and Alzheimer's and the neuropsychiatric disorders. Having a system that reliably recapitulates function, whoops, there's Fred, is going to be incredibly useful. So Fred.
That's the first time I've heard, whoops, there's Fred. Yeah, hi, Rusty. It's been
Hi.
a very long time. And so when you were describing your model and its separation from a vascular system, I'm interested of whether you've looked at Clive Svensson's work, who's done a lot of work in this area also, but where they've now created sort of what they call a brain on a chip, where they have the vascular system, they have the neurons and the astrocytes and all of that, and they can watch the interaction. I'm wondering how that work informs, you know, the future of what you're doing.
Yeah, I have been following it, and we have a separate effort using, it's not a chip, but it's, you know, microfabricated tool. We're working with the engineering department. And we can get, what you have to do is you have to flow, you know, we make our vascular organoids and they will penetrate into the organ. Their survival length and duration is limited, and you're relying on providing artificial CSF through and blood vessels through the blood-brain barrier is not so sturdy in those cases where you can sort of test organoids, but I'm hopeful that technology will come along and will move along and complement the transplantation work as well. I would say though that ironically, you know, this model is going to be dependent on where you put it and how old the animals are when you do the transplantation. This is not addressing your point exactly, Fred, but I hope I address that. I don't feel like any of us are in competition. I feel like we're all striving towards it. And if you can get an in vitro system where it actually has an intact blood-brain barrier with parasites, and they penetrate and they are human, and they fulfill the obligatory nutriments that are necessary to keep them alive for extended periods of time and mature the neurons, then that's great. I think that'll be a good complement to what's going on. One interesting thing is that this model that I presented to you was actually developed. I developed this in 1984 when I was in Sweden, and we were trying to find locations in the brain where we could transplant superior cervical ganglion to test whether or not any different areas of the brain had nerve growth factor, human nerve growth. This is a long time ago. So we implanted it into this colliculus, which is a budding medentate gyrus, actually. But we tried lots of different areas, and there are very few ports in the area where they survive for extended periods of time and are highly vascularized within the tissue. We're still exploring other areas, but I think that's an issue that has to be considered. The other issue is the age of the organism. If we transplant early on during early development, then the organoids tend to grow much larger. And we've chosen to graft into the adult, so it restricts the amount of growth that the organoid will go through. But remember, in the developing brain, the brain is growing at the same time. And a lot of the factors that are involved in inducing its own cells to growth are impacting on the host as well. Another, since not more questions, another interesting fact is that if we don't put astrocytes or microglia in the organoids, then the host microglia and astrocytes will migrate in. But if you put human microglia into the organoid, it prevents you'll see the microglia on the brain side, on the mouse side, but none of them co-stain with human markers. And that's true for astrocytes. So there's an interesting evolutionary barrier for astrocytes and microglia that will not penetrate into the human tissue if the obligatory or their sister cells are present.
Okay, final question, Abla.
Thank you, Dr. Goldstein. Again, thank you, Dr. Gage. Very nice presentation. I wanted to know, just philosophically, this beautiful system, can it be used for the study of biology and the pathogenesis of diseases of the brain, or is it mainly a screening methodology for potentially identifying agents that affect each of the cell types?
I want to make sure I understand your question.
If we are interested in the biology of understanding mechanism of disease of the brain,
yes,
is this, do you think this will be a good system to do that?
yeah. Well, so we've shown, for example, that I'll give you two examples. In vitro, in monolayers, we've shown that bipolar cells, hippocampal bipolar neurons from patients with bipolar disease that are non-responsive, or say that are non-responsive to lithium, tend to be hyperexcitable in a monolayer setting. But we're restricted in terms of how much mechanistically we can understand that. And while we do see this hyperexcitability in the organoid setting, and we want to understand whether or not we can use this as a tool to get a better understanding of that that hyperexcitability that you see in the lithium non-responding patients. So we certainly believe that the organoids will be a vehicle for understanding pathophysiology of disease, and that's our main goal in doing this. Is we also have another study in depression where we find that patients that are responsive to ISSCRs have a different profile than those that do respond to ISSCRs. And again, trying to nail down the mechanism for how that pathophysiology is manifested. So we believe that this is a good model for tracking that down.
Thank you. I was mainly thinking about what are the triggers for the pathogenesis of disease and how the disease progresses. But it appears that eventually you have all the cell types that you need in order to answer that kind of questions in that organoid. Is that right?
Yeah, and I was sort of hoping that one example I showed you where you take the neurotypical microglia and put it into a diseased organoid and show that it is the host that's activating those microglia suggests that the pathogenic signal to get an inflamed brain is coming from the host. And we are looking into what features in the host rather than concentrating on the microglia. What is the host doing to activate and aggravate these microglia?
Great, thank you.
Yeah, thank you very much, Rusty. That was really edifying, exciting, and I think it's going to make a big difference moving forward in the coming years to have models like this and others that are being developed. So thank you very much for your time. And we are at a transition point. Rusty, you're welcome to stay as long as you want. But our next topic...
Rather than I leave?
No, it's totally up to you.
I've got 8 minutes for my next meeting, so I could hang on.
Good.
I
Hang in
also
there.
want to thank you all for inviting me, and thank you for your service to the committee and to the community for serving on this board.
You got it. So next up is Vice President Rosa Canet-Avalos, who will present a proposed concept plan that if we sign off on it, we'll then move to the science subcommittee, and then if it does well there, on to the full board. So Rosa, please take it away.
Thank you, Dr. Goldstein. And, this presentation from Dr. Gage was very on point with what I'm going to present, as it was furthering the evidence of the utility of human stem cell models in modeling the pathology of neurodiseases, and especially the importance of the neuroimmune axis, for example. So we will hear about this in a few minutes as I go along. So this is an opportunity that we have to present the first phase of the CERM neuroscience strategy implementation, and it will come in the form of the neuro discovery concept, also known as REMIND, and REMIND stands for Research Using Multidisciplinary Innovative Approaches in Neurological Diseases. What I will be presenting today is actually a concept that will start with a pilot. And this is what we are proposing is a phased approach that I will be explaining as in later slides. And we are going to pilot this as a potential framework for multidisciplinary discovery research at CERM, with a growing investment adapting to CERM's growth in other parts of its infrastructure. So I want to clarify that this is not only for, as you will see later, neuropsychiatric diseases. It's about disease mechanism research, and we are going to pilot this with neuropsychiatric diseases, or that's what we are proposing. But if the model of this initiative is successful, we will be growing into other diseases. I wanted to make this clear so that there is no confusion. So let's get started. This is CERM's neuroscience strategy has been developed in the context of our mission statement, and it maps out and integrates with the elements of our mission and a strategic plan, and specifically with the first theme of our strategy, advancing world-class science, and the two main goals, which is develop competency hubs and building a knowledge infrastructure, or the knowledge networks. The REMIND initiative corresponds to the discovery phase of CERM neuro strategy, and the TRAN and the CLIN will be addressed separately. I also want to make sure that we provide the right context for today's discussion. This is all a reminder that this comes from within the context of Prop 14's mandate and CERM's $1.5 billion set aside for mental health research, with the potential to transform the treatment for diseases and conditions of the brain and the CNS. The goal of this slide is to provide a frame for the background and the rationale for the conceptualization. For the conceptualization of the current concept, and the vision of the neurodiscovery strategy has been informed by multiple layers, as you can see here, of a stakeholder discussion and input that started even prior to the passage of Prop 14 over the past 2 years, and is outlined in this timeline chart. And there are three major takeaways, key takeaways. One is that there are major gaps in our understanding of mechanisms underlying disease processes in the brain. For example, we know a lot about how the heart works, and which has been key actually for developing therapies, but the brain is far more complicated, as we've just seen with Dr. Gage's presentation, and we know very little. So that has been impeding the progress in finding therapies for people with mental illness. And the key takeaway is that the lack of understanding of these underlying mechanisms of disease processes in the brain is a major bottleneck in the development of successful therapies. Now, in order to discover these mechanisms, one of the best ways is to leverage collaboration, right? So the most effective and productive way that we heard was the development of a consortium approach where genomics and big data, novel stem cell models, patient data could be collectively leveraged to advance the field of neuro research in a collaborative manner. And in order for a consortium to have its maximum output, we need to promote knowledge sharing and expand shareable resources to accelerate research of complex diseases. So what has CERN done to cover this lack of mechanistic neuro understanding since its inception? For that, we needed a gap analysis of the portfolio. And in the past neuro task forces, we provided this portfolio gap analysis. At the last task force meeting, we presented an internal portfolio gap analysis. And this slide summarizes the historical funding for discovery, which was up until now $1.2 billion. The neuro-funded discovery, 28%. And of those 28%, there was a 4% in neuro disease mechanisms. Now, that wasn't a lot, not significant. Where did the other 24% go? Well, it went partly to fund scientific progress, refining differentiation protocols, and creating more complex models in a dish, such as organoids, as we heard, so that the field could be ready to study disease mechanisms. So CERN invested in the infrastructure and the basic foundational research of this to make the field ready to study with the models. And that's kind of where we are now. What is the focus that we are going to have? The focus that we are proposing is generation of novel therapies for neurodiseases, which requires uncovering the underlying mechanisms. Therefore, the first goal of CERN's neuro discovery strategy, which corresponds to the goal of the concept for the program that we are proposing, could be to accelerate the discovery of mechanisms underlying CNS disorders, leading to the identification and validation of novel targets and biomarkers, with the goal that these efforts would provide new avenues and rigorous foundations for other translational and clinical development work. And as you can see, I mentioned here, neuro is not neuropsychiatric. What the goal is the goal, remind, is a concept for a large initiative that could be phased. The implementation of its first installment, we are going to propose to be neuropsychiatry, but it's not going to be all. So how are we going to go about this? What are the specifics of how we will go about achieving this goal? The objectives proposed for this initial program are to first advance foundational scientific understanding of neurological and disease mechanisms. The goal is that these efforts will ultimately provide new avenues and rigorous foundations for other translational and clinical development work. The second would be to catalyze multidisciplinary innovation and attract new talent and ideas into the study of neurodiseases. We need to incentivize and catalyze an open collaborative science ecosystem and support this kind of interdisciplinary collaborative teams, empowering the next generation of scientists and bringing together outstanding, innovative, forward-thinking scientists from different disciplines into a collaborative network. Ultimately, multidisciplinary teams with big data, computational analysis, focused discovery work can lead to the identification of novel targets and biomarkers with immediate implications for clinical trials. And this goes hand in hand with incentivizing an open collaborative science ecosystem through data and knowledge sharing infrastructures. Now, another objective derived from this collaborative environment is to motivate and support innovative and bold transformative new ideas and tools that address fundamental challenges in CNS disease biology. A very good example of this was, for example, optogenetics. If we can investigate how the neurons work together by using light to turn some neurons on and record the response of other neurons, we've advanced a lot in the field. So if we had invested in this, that's at CERN, that could have made a big transformation funded by CERN. So the last one is to leverage and connect with CERN's existing infrastructure of programs. And we will see this as we show how everything maps together within our ecosystem of CERN-funded programs. But we are talking about the shared resource labs, the competency hubs, infrastructure platforms like the Data Coordinating and Management Center that will be, we are conceptualizing it right now, and others. So what are the opportunities that CERN can leverage to put this goal in place? The first one, as we mentioned, is the $1.5 billion, and just as it's not all that would go to just the neuro discovery, obviously, but part of the Prop 14's $1.5 billion earmarking set aside for research in mental health, CNS diseases. The second could be the scientific extent, innovation and expertise in genetics and stem cell biology and neuroscience in California. We have a deep pool of California stem cell researchers, including some supported trainees and investigators. The world-class California stem cell research infrastructure, including CERN-funded shared resource labs, the IPSL Biobank, the planned data infrastructure, and others. Also, large amount of data and resources from other neuro-focused consortia initiatives, and advances in stem cell technologies to study the entire diversity of Californians with diseases of the brain. And as you will see, some of those advances have been pointed out by the task force meetings. Now, in order for the collaborative research to have an impact and accelerate our understanding of these diseases, the scope of the first initiative should be focused. There are many neuro diseases with multitude of mechanisms, and these mechanistic worlds do integrate, but we need to start from the bottom with a focus. And in order to do that, the board requested that we do another gap analysis. And we found that when we mapped the discovery research from CERN's inception by disease to the disease burden in the US at the time, we found that neuropsychiatric diseases had not been funded by CERN. So basically, neuropsychiatric diseases were historically underfunded at CERN despite the large burden and unmet need. So we propose that this could be a good place to start. Therefore, the Neuro task force started with a series of meetings that made the case that the neuropsychiatric space was primed for rapid progress due to several recent advancements. One was that genetic risk architecture was starting to be defined. It's still far from diagnostic or predictive, but we are getting closer. And we are getting better at translating loci to genes to pathways. The second point that we heard about was the demonstrated utility of human stem cell models. And we heard a little bit more about this today. Basically, mouse models have revealed complex interaction of genes and circuits and behavior, but they have severe limitations. For example, they capture poorly the impact of non-coding variants, and we've learned from Kristen Brennan's and presentation that it's not ideal. There's also the advancement in related research technologies. What can they teach us about psychiatric disorders? And with all of this evidence that we heard, the focus of the first implementation for this concept we propose to be neuropsychiatric disease mechanisms. So how did we propose the structure of this RFA program to fund acceleration of discovery of disease mechanisms in neuropsychiatric disorders as the first phase of this pilot program? So for this, we are proposing two types of awards. A first type of award, the large collaborative research projects, could require data, preliminary data. It could be a 4-year award with a base component of $2 million per year, with $8 million in total over the 40 years. We would expect a number of 6 awards to be funded with a total budget per cycle, which we will see now, and about 5 or more investigators, minimum of 5 investigators. Now, we thought that we would like to incentivize collaboration, and to do that, we decided that if we could, if the teams bring matching funds of a minimum of $0.5 million a year, this can be from industry, from outside collaborators, from other consortia, and it does not need to be from outside of California. It can be from inside of California. So a group could be collaborating with a company. So if you bring $0.5 million a year in funding, CERN will match those funds with a total of $2 million in total for the 4 years. So this led to a total for this type of program of $2.5 million a year, which could be 3 with the matching, $10 million in direct funds cost per award for a total of 4 years, and a total of $72 million with indirect cost as the total that we would be that we had asked for this program. The second type of awards could be more exploratory projects, more proof of concept or initial validation of a proposed tool, model, hypothesis. This could go with the innovative part of the objectives. This could be without required preliminary data, 2 years, and $0.5 million a year with $1 million total for the award. 15 expected number of awards, $80 million in total. And this could be 2 or more investigators, a minimum. Now, how do we see this flowing through to the timeline? So REMIND-L could be 4 years. The large collaborative programs could go, and then there would be the opportunity for 1 more time renewal for 4 more years in the next cycle. And in cases where things had advanced that can be leveraged by a TRAN or CLEAN, there would be the possibility of dynamism to this mechanism that we will explain through discovery advisory panels. So for example, if you are in year 2 and you have a discovery advisory panel, you've already shown validation of a new target, you could actually move toward a translational award or application of some funds. And then REMIND-X could start in year 2. So we would have an RFA starting, posting next year in 24, and then in 25, we could have REMIND-X starting again. Now, this is a pilot. Again, what I wanted to show with this slide is that we are piloting a new framework. And these awards that we would have, the exploratory with the larger collaborative awards, and then all interoperating with the discovery programs, all of these could be repeated we would have another, and I'm not saying that we're going to fund specifically these are only examples, but then we could have another one in 4 years' time that could then start with neurovascular and neuroimmune axis type of focus of disease mechanisms, other neurological diseases, other focus areas of bottlenecks. What we are trying to show here is that we are trying to pilot a framework for a way of funding discovery research in neurological diseases. This program, as I was showing, we are proposing a phased approach that will allow a work continuity and expansion to other areas of CNS research, piloting this potential framework for multidisciplinary discovery research at CERN, and growing in investment, adapting to CERN's growth in its infrastructure knowledge network and competency hub capability. Again, this is not only for neuropsychiatry. Neuropsychiatry would start this year with what we are proposing, $72 million and 6 teams, and then 15 teams and $18 million for the innovation. But then it could, as we move to the next round, we could still include neuropsychiatry, but then we could fund other diseases. These are examples only. And we could allocate more funding as the infrastructure would already be settled and it would already be interoperating with the Data Coordinating Management Center and other parts of CERN's infrastructure. And we would have already learned how to do it with other consortia. Then we would be able to fund even more, and we would be growing, and we would be able to figure out common mechanisms amongst these diseases as well. And then as we grow even further, we would be funding more. The next slide. Actually, I forgot to make a point here. In here, you can see that this phased approach could be also coordinated. The programs could be coordinated, adding a discovery advisory panel. The discovery advisory panel could be providing input to the researchers, to the awardees. So this is a part of the program that CERN could put in place. Same as we have translational advisory panels and clinical advisory panels, we propose to have a discovery advisory panel that is some experts that will provide input so that we can learn as we move forward and help the awardees leverage each other's research, and in some instances, if things are advancing faster, to help them move faster towards translation or clinical. We could also have an annual network conference, and all of these data could be working and interoperating with the Data Coordinating and Management Center. Again, this slide is the one that shows how this program budget could be growing as we are growing the program and as new diseases are coming in place in this initiative. And then this slide also intends to show what the estimated projections of money spending could be for this program. So the CNS projects are, this is for the Remind. So it could be $72 million. Then if we make 12 teams, in 4 years' time, 144, 12 more teams, $144 million. We could keep about the same level in the Remind X. But then we also need to take into account the CNS projects that are part of our DIS 0 and 2 pillar programs. Currently, as we are making changes, concept amendments, we are going to be increasing the amount of funding to these pillar programs. And this is an estimate because this has not yet been presented or approved by the board. But imagining that we end up funding at the level that we believe might be funding, this could correspond to about $235 million in the next 12 years, which means that the total discovery neuro funding approximately could be about $648 million of the $1.5 billion earmark. So this is just a rough approximate. Again, this is a presentation of an overall concept for a new initiative, a new way of funding neuro discovery science at CERN. Again, a reminder of the key program drives, the REMIND program drives key objectives to accelerate foundational scientific understanding of neuropsychiatric, but in general, neurodisease mechanisms and the development of novel tools, catalyzing multidisciplinary innovation. You've seen the structure that we are proposing for these large teams, attracting new talent and ideas into neuropsychiatric research, and seeding new partnerships. Just as a little bit of information, we've been talking to different partners, especially the federal government that are funding, but also other partners, so that we can leverage all of these together, all of their investment as well into this initiative. And then driving open and collaborative science and aligning best practices through data and knowledge sharing infrastructure, which is something that we are working very hard, and that we will be providing a concept. So this could be implemented right after we initiate. And we are taking into account how we could make it work, given that the data coordinating and management center could happen right after. This could all be coordinated through the data coordinating and management center steering committee. And again, this is just a representation of what this model is taking into account in terms of programs, the large research teams, the early projects, and the CERN discovery pillar projects. And checking with time, Larry, am I doing well? We have a few more slides, about 5 more slides. We're okay?
That looks
Okay.
fine. I'll leave some time for discussion, though.
Wonderful. Yes, about half an hour. So the REMIND high-level outcomes could be the novel mechanistic insights. So REMIND L, which is the large collaborative program, would lead to novel mechanistic insights into the biology of neuropsychiatric diseases, could allow us to get further understanding of current mechanisms, including mechanisms cutting across classically defined disease boundaries. And as you can see, as we add more diseases, we will have a chance to find those common mechanisms even more, but we need to start with something defined. An extension of validation of findings to diverse human populations, as well as identification and validation of new therapeutic targets or biomarkers. And then REMIND X high-level outcomes could be to provide us proof of concept or initial validation of proposed tools, models, or hypotheses. So this is a model of how we see it all working. Ultimately, this is a multi-dimensional and layered proposal that pulls different components together in service of the overall neuro strategy, and consistent with the feedback that we've received from the members of the board and the task force over the past few months. The goal is to accelerate the pace of discovery and inform new paths to cure neurodiseases, leveraging in already existing infrastructure. As you can see here, there's the discovery, the shared lab infrastructure, even the training, education infrastructure, that then also leveraging external consortia, resource networks and data platforms, and ultimately leading to this open science community ecosystem that will lead to discovery of novel targets and biomarkers and increase the efficiency and success of clinical trials. That's where we are all trying to lead to. Now, in terms of project eligibility, to be eligible, REMIND projects must propose studies that are focused on elucidation of mechanisms of neuropsychiatric diseases. That's for the first installment of this program again. So this could be the concept that we could come in September with, that would be the RFA, but as we move in 4 years' time, we could be adding other diseases and include the studies using human stem cells or genetic research. Note that any studies using non-human systems must be validated with a relevant human cell equivalent. In terms of principal investigator eligibility, for both types, we are California, all principal investigators should be employed at California nonprofit or for-profit research institutions. There has to be one PI that's going to be designated as the coordinating PI, who will manage the collaboration and will be the administrative contact for CERM and any grant partners. The minimum % effort for the coordinating PI in the REMIND-L, the large collaborative, is 20%. And for the REMIND-X, which is the exploratory high-risk projects, is going to be 10%. Other PIs, we're asking for the 10% minimum. The team size, 5 minimum for REMIND-L, and REMIND-X is 2 minimum. And for REMIND-L, we are asking that at least one member of the collaboration should have relevant clinical expertise, and one member should have relevant computational biology expertise, given the need to link to clinical and also to be able to leverage the data. And CERN will encourage favorable consideration of applications that include at least 1 to 2 early career faculty. For the REMIND-X, we strongly encourage applications from investigators who can bring new technology resources or frameworks to the study of neuropsychiatric disease and in vitro modeling of CNS. Now, in terms of data sharing, one second, it got stuck. All proposals will need to include the data sharing and management plan and describe an approach to sharing and management of the data generated consistent with FAIR principles, findable, accessible, interoperable, and reproducible principles. And it also must coordinate with the Data Coordinating and Management Center that will be presented the concept in March of 2024. Now, here, there is a timeline situation because the DCMC is coming later. But given that this RFA, if approved the concept, the applications could be reviewed in May of 2024, we will be creating a process by which CERM will meet with the awardees during pre-funding administrative review to make sure that their data sharing and management will align with what we need for the Data Coordinating and Management Center. So we've been thinking about how this could be doing, and this has been also advice that we gathered from colleagues from the federal government that are doing similar initiatives. And then diversity, equity, and inclusion, the applications need to include plans to address DEI. Okay, the data advisory panel, discovery advisory panel, CERM, this is very important, will coordinate data discovery advisory panel that will be composed of non-California experts to provide independent, confidential expert advice on REMIND programs. This is what I was trying to say earlier, but this is a bit better articulated. The specific activities of this committee could include review of the programs reported by these large collaborative awardees and provide non-binding advice to the awardees and CERM. So basically, we will provide this as a resource, expert resource for our applicants. And this from our colleague Abla Crisi has she has told us that it's an extremely helpful resource for the translational and clinical researchers. And that also will help us identify and leverage external resources to further collaborative research. Now, the budget. So the overall concept, we are not asking for money here. This is just the estimate of what we would be investing on the first phase. We are only giving a projection of the funds that will be required because the budget for each one of these RFAs is being asked separately at the corresponding joint ICOC. So the budget for the Remind L was included in the Discovery budget for fiscal year 23-24 that was presented by our colleague Pune Sinsam at the last June meeting. So what we are asking is the request of the board to approve the proposed Remind program concept as an initiative that will help us further the discovery of disease mechanisms in neuro diseases, and that we will be implementing in its first phase with neuropsychiatric diseases as a focus. Thank you.
Thank you, Rosa. That was a terrific presentation. So questions and or discussion from the task force? Steve.
You're muted, Steve.
Yes, wonderful presentation, Rosa. Thank you very much. Can you go back to slide 15, please?
Yes.
Hang on just a minute.
Yeah, just give me a second. I forgot to. There you go.
No, that it's the next one, or the one before it.
Yeah,
the slide.
we will just this one.
No, it's the one that shows the use over time of expanding the program. So I counted that as
yes.
slide.
Yeah,
Thank you.
no, it's there's so many.
That's it.
This one, correct?
Right. So this is a more of a question, I guess, globally, and maybe for Maria Milan. But this suggests that we could be funding these programs on through 2035. And we have this neuro budget of 1.5 million, which leads then, what, 4 million for the remainder of things that CIRM does. Do we have a timeline plan for the existence of CIRM outside of the CNS area? In other words, is CIRM going to be around in, as before, when we were thinking about what was going to happen with Proposition 14, and it was a very close call. We had to have a plan of what would happen if we didn't get refunded. And I think that's always wise to keep in the back of our mind that, the third time may not be the charm. And so the integration of spending $1.5 billion in the CNS area and the existence of CIRM writ large spending money otherwise, I don't know, is that being thought of as we go along so that we're going to keep CIRM going independent or in conjunction with the CNS area on up into 2032, 2035?
Larry,
Sorry, it's complicated.
Larry, did
Maria
you want me to
Milan, can
respond?
you?
Sure,
Yeah, please.
absolutely.
Yes.
Right. Thank you so much for that question, Steve. So Pune Simpson and the team have been since the passage of Prop 14 have been deploying a forecasting tool in terms of expenditures over time, both for the research and administrative budget. There are a variety of different models to that, but some of, let's say the base case is an expenditure of what the maximum allowable funding is according to Prop 14. There are some, some exceptions to that. And then also calculating into it return funds, et cetera. And so the timeline that Rosa presented is compatible with the projection in terms of the administrative runway according to this modeling, as well as the research runway with those funds. So there's kind of these parallel type of forecasting. It's also, this forecasting tool is fed by what our actuals are, our actual performance is. So it's a pretty, the assumptions that are built in with a very aggressive, expenditure of the actual budgets we budget per year. And as you know, there's variance. So she has a bunch of different models. So I hope that answers your question. It's not, so it is the timelines that Rosa presented are reasonable to those projections, especially a 2032 timeline. And so that's kind of, hopefully that's responsive to your question.
Yes, it is. Thank you, Maria. I think, it's something we just need to keep an eye on as we go forward. My worry is that, we'll get to a point where the only funds that are left are for this area, the CNS area, and we don't have any funds available for funding other projects. And then how do we run the organization at that point? But that's a long ways down the road. But I think it's something, as we move along, we need to keep an eye on, because we have a huge research budget, this time around. The approval was, up close to $500 million, something like that. So that's going, if we spend all that, that's going through money at a very fast pace.
Absolutely. What Rosa presented in terms of what our historical expenditure have been on our pillar programs, given the percent of those kind of organic programs that are coming in, she presented an estimate of how that could still be funded. And then I think Rosa's right here, so I'm saying like she's not in the room, but she's right here. But I think that with the estimate of these mega programs that she's presenting is I think at least 4 or 5 of these big, consortia type approaches could then be funded not only for neuropsych, but other types of neural fields. And then in total, that would compose a 1.5 billion, in terms of the earmark for neuro, for CNS, the combination of the organic things that come in through the pillar, plus the special program project or consortium. And then the remainder of the funding would be then available for other non-CNS types of initiative across the different types of research programs.
Good, thank you. Great question, Steve. Pat.
Yeah, thanks. Rosa, that was great. And I love the concepts. I want to talk a little bit about or pose issues around sort of the content of Remind L and Remind X. I love the concepts. Can you go to the slide where you've got, I don't know what the number is. It's the slide that has the requirements in terms of investigators and effort. And if you can go to that.
Yeah. Let me see. Principal, is this the one?
Yeah, that's good. So the principal investigators for the Remind L, you want one clinically, one who has clinical expertise and one who has computational expertise, right?
Yes. 1 second.
Okay.
My
that's fine.
My mouse is
that's good.
cutting.
So I just need more clarification about what you mean by the team. So when you talk about 5 PIs, for a model like the P50 at NIH, a PI is a project, is these are all integrated, of course, around a specific theme or hypothesis for discovery. Is that what you're talking about here? 5 projects, and one of those 5 would be the coordinating PI. Or you talking about 5 investigators, some of whom might be serving a purpose of general leadership in a clinical area of focus, and one would be the serving in general all the projects that have computational components to them.
And we are talking about, thank you, Pat, for the question, very relevant. We are talking about not 5 distinct projects. We are talking about one large collaborative project that can have different aims that different people might be working on, different PIs. There should be one PI that coordinates and will be the administrative point to the award with CERN, and it will be the person that will be responsible for, if we have a discovery advisory panel and there are something that needs to be implemented, he will be the responsible to coordinate things. He will be the one that will wrangle everybody together towards making sure that we get to the outcomes, the goals that they put. It doesn't need to be, we don't need one lead that's clinical, one lead that's computational. What we want is the expertise. It could be that the other PIs, so we have 5 PIs is minimum. It could be that there are 7, no, but 5 we think it's a good number for this kind of collaborative projects. It could be that one of the other PIs, the 10% is a clinician of the disease that will be the one that's going to bring the relevant type of questions that are relevant to the disease mechanisms that will be discovering for the clinical trials, right? And that will also be the context to the patient populations. We need to have the end in goal, and I think having a clinician in the team is very relevant. The computational biology expertise has to do with being having a computational biologist will make sure that there is some voice there that keeps everybody thinking about what metadata do we need to gather, what are the standards that we need to have, who do we need to talk. So that's what we want to have, right? Those two expertises are key.
Yeah, that'll make sense. This is about the architecture of this, which I think is really important so that investigators are clear about it. If you've got collaborations across sites, it means each site is going to have a statement of work, a set of responsibilities that they have with separate budgets, right? It's not. It's unlikely what you're looking for. Is it likely that it's going to be at one institution? This matters because of how you would structure this, and so the reason there's a theme around a program project grant that has a singular theme, and then there are elements to it. One might be expert in imaging, that's going to be done. One might be expert in electrophysiology, et cetera. But they all address the core theme, the core question. And it sounds like here you've got one large project that's going to have multiple PIs to it. But if there are different institutions, the logistics of dividing that pie the way that PIs are used to operating is going to be an issue. So I think you may want to think about how you want to structure this so that it's clear what CERM is looking for. The other is that, and we heard this from several presentations, 20% effort for, let's say, a senior scientist who has the ability to coordinate a program like this is a high percentage, and it's likely to eliminate a number of individuals who we know, some of whom presented to us, right, where they just don't have the effort, and as you know, legally we can't go over 100%. Of course, we all do go over 100%, but we can't legally go over 100%. So one thought that had as going through the slides is a coordinating PI would have to certainly designate 10%, but the other PIs might be at 5%. And there were like, from what I can recall, at least 3 presenters who talked about the effort requirements. I think for the RemindX for the discovery phase, I don't think 10% is an issue. I think those are fine. So think about that. I think the dollar amounts are really aligned well with what investigators are used to when they're putting together a program project, P50. And I think I've mentioned this to you before, when in different institutes at NIH do it in different ways, but when they do their analyses of return on investment, these programs generally do really well because they're structured and organized well. The other thing to think about is for the advisory panel, and that was singular, so I'm assuming an advisory panel, but you're looking at 16 projects here, and that's a lot.
It could only be for the large ones. The smaller ones don't need an advisory panel. It's for the RemindL. It's the 6 projects.
So I'm just saying from experience, they're there to just serve when somebody would send them a question, or they're meeting to review the progress of the programs? Are they there to provide CERM with feedback and how things are going, or are they there just to be expert sources?
We will use them mainly for a yearly progress review and provide advice to the grantee, to the awardees, and to CERM recommendations. But I think we will also use them ad hoc. We might not have all the expertise internally. And if we need to use them as consultants for certain questions, when we need to maximize the outcome of these research projects, we will. So that could be the idea.
Yeah. So I don't know how some of us on this call have served on these kinds of scientific advisory boards, and 6 projects of this size is a very heavy lift for a single committee to do. Even if you could have a large committee, and then each person gets 1 or 2, but
it's not one committee. It's a discovery advisory panel. We are going to have it's like the clinical advisory panels or the translation advisory panels. It's not the same 3 people for all the awardees, for all the awards. It's a combination. So we'll have a pool of, say, 15 expert consultants, and we will mix and match depending on what's the project that we are looking at. So probably each one of them might have a maximum of 2 projects that they will be advising on.
Okay. One thing to think about is that for all P50s that I'm aware of, they have to have a scientific advisory committee, and they're the ones who define it. The scientific advisory committee essentially does what you just said. They meet annually, and that's another model where each of the large programs would have an advisory committee that would send a report to CERM. And they may be as or more effective because then you have the investigators having some input into the expertise that they feel is going to be most important. The only thing I just wanted to mention, I have some wordsmithing that I'll send you. I'm not going to bring it up now. It may be worth thinking about, particularly for the large projects that have, it's just the bridge with, clinical disorders, having, in addition to a DEI plan, a community advisory committee may be worth thinking about. CACs are really helpful in thinking about, particularly since a lot of this involves, patient material that is being used, et cetera, and having a CAC, a small CAC for the programs might be really, I think, important. And we have patient advocates on our board that I think may feel the same way. For us and my own projects, it's been incredibly helpful to get insight from a community advisory committee that's relevant to the studies that we're doing. So that's something to think about. Thank you.
Thank you, Pat. So let me just catch something. When you said the PI commitment, what were you suggesting for Remind L? I didn't catch.
So every P50, which is the NIH term for what you're describing, has a scientific advisory committee. It's usually 3 or 4.
No. The PI, the percentage of the PI.
I was
did you end up?
I was going to recommend, based on the feedback we got and based on my understanding of where, this is going to draw. CERM wants to draw the most impressive, scientists into this program. We heard from one today. Does he have 20% to commit? He's not on the call now. I'm not asking him, right? Even if he was, I'm not asking him to reveal, but 20% for that kind of a laboratory for the PI is a lot to ask. And I don't know how others on this, on our task force feel, but if that was.
Could it be that we allow, we are asking for someone that's more new to the field, but it's a PI, that be the coordinating PI at a 20%, and then the more I'm just thinking, we are asking for at least 5 people, so they could. We can discuss. We can discuss that. I hear you, and we were the same as you. 15%. Could that be fair? 15% a coordinating PI.
So if you have 4 at 5%, let's say you have 2 at 10% and 3 at 5%. That's 20. That's 35%. That's 0.35 for a faculty commitment. That's pretty substantial for a program. I look at it as a sum, not as individuals. And I don't know, the group will have to contemplate this. I have my own views that 10% for the coordinating PI. And I would say that it would not be a good idea to have a junior investigator to be the coordinating PI.
Might not be the coordinating. Yeah, I know. I hear you.
It's a lot of work.
But,
It's a lot of work. It takes some juggling to do it well because there's always conflicts that arise. Not bad conflicts, but there's always issues that arise, and I think it's challenging for a junior person to do that. But I should let others speak.
let me see. Yeah, I was just trying to see
Great. So
in comparison with
Pat.
other. Okay.
Rosa, we'll have to continue this conversation offline, I think, but it's an important point. Fred.
Thanks, and I won't belabor it, but I think this slide, I really want to thank Pat and Steve for kicking this off, because I think they also started where I wanted to start. It isn't clear, but I'd like to see, not today, and I don't even want to hear about it today because there isn't time, but I want to understand how the staff and any others involved in putting all of this together actually took in the reason why there's not more neuropsych money being spent. It's not because they don't know about us. It's not because they don't want the money. What we heard was it's too much effort for too little dollars. So how did the CERM team take that feedback and build it into this plan so that in the end, we don't find ourselves looking at another pie chart that says, oh, neuropsych was, still underfunded, and we put this whole great program together because we didn't pay attention to the feedback we got from them that it's too much work for too little dollars. Again, don't want to take the time now. That's one. If you could go back to the slide that Steve talked about that showed all the arrows and the spending and it's after that, because it gets down to a total of barely, yeah. So what's not helpful about this slide is you're talking about a neuropsych program, and so do I understand we're talking about 240 million out of 1.5 billion? Is that what we're talking about? Spending on neuropsych?
No, as I mentioned in the previous slide, let me just show here. What we were proposing is that we would be starting with neuropsychiatric as a pilot, but 1 second. As we move forward, we would be including other diseases and more working focused on systems. So research mechanisms, disease mechanisms of the neuroimmune axis, and include neurodegenerative, neuropsychiatric, neurodevelopmental, for example, neurovascular, the same. Those are examples, but we could we could increase the scope of diseases by focusing on systems. That's how we were proposing to move forward. So no, the 678 or $48 million could actually be for these neurological diseases, discovery of disease mechanisms, neurological diseases. Only the first 4 years could be focused on neuropsychiatric. Fred, thank you.
So Remind L and Remind X are not exclusively neuropsych. They are,
Correct.
this is what you're calling this whole expansion. And now my question is, where did these other indications come from? Because to my knowledge, they haven't been discussed by this committee.
No, and
These are the things that we should be focusing on in future years.
correct. And we would be coming in 4 years or 3 years time, as we are moving towards the next set of RFAs, we could come to the board with the specification of, we could come with an outcomes analysis of what we have done with the money of neuropsych, and what we think that might be best to leverage it with, but up to you to decide where you want us to focus based on whatever analysis you want us to do. So this is just to show that the Remind L and X, this Remind concept structure is for discovery in neurological diseases. And we are going to apply it first to neuropsychiatry, and then we will come to you to tell us what else you want us to start bringing in, but giving you outcomes, giving you what is it that we've achieved in the next 3 to 4 years.
Yeah, so you have to take off any other disease indications. Call it indication X, indication Y, indication Z. When you start putting things in writing, you create the expectation that that's where this is starting. Just like it still remains a mystery how we ended up starting on neuropsych, but it is what it is. And when you start putting things down, it seems to have the setting in stone function. So if you're just talking about adding disease indication, have it just say literally disease indication number 1, number 2, number 3 to show it growing. So and what you started with here was we're looking at a program that apparently has a subset of costs. Now, if you click on another slide where you get to the 648 million, that we get to 648 million of total costs for this new initiative, which is 43% of the total minimum for neuro. Given that my understanding is that CIRM has already spent at the prior funding cycle 1.5 billion, or maybe it did, or maybe it didn't, I don't know, and I don't want to rehash this, but I need to understand whether we're actually going to not be able to fund things that we would want to fund because we're looking at carving out 43% of the 1.5 billion for neuro, which I want to know what that total is for neuropsych, plus some other things. So this slide where you have these arrows, like, it's misleading because you're talking about Remind L and Remind X, which in these future years is going to be something other than neuropsych. But what you've put under the arrows, I think, is funding strictly connected to neuropsych, which
it's fun.
Well, it's 12 teams, 144 million. You're talking about neuropsych.
No. For Remind L, it could be for different diseases. It could be that in years 2028 to 2031, we decide that what CIRM is going to fund is research around the neuroimmune axis, disease mechanism across all neuro diseases. And that could be the funding. 12 teams could be really large because those are $2.5 million a year teams. So it's like $10 million a year award, $10 million awards, 12 of them. That's a lot of money, right? That we could be funding. but it's not neuropsych. It's to do with all neuro. And I took your point that we need to remove from the slide 17 the indications. We are starting with neuropsych now. The total will be $168 million for neuropsych, but the 648 million could be for all neuro diseases and discovery. So this could be what we are asking for the neuro strategy at CERN. The discovery part, we estimate that might take about 42% of the funding, including, what we will spend in the DISC pillar program as well.
Well, if you want to know more about the confusion of this slide, I won't take the time here because I see Leandra has her hand up. This is a very confusing slide. I don't understand what the salmon arrow is at 235 million. And if I understand what you're saying, the total commitment of 1.5 billion dedicated to neuropsych is 168 million, which is 11.2% of the 1.5 billion. That's what I want to understand, because when you're talking about starting up with a pilot around neuropsych, the question is, how much are you going to devote to that? And the answer is 11.2% based on what you're telling me today, but I could not derive that from any of these slides. And then there needs to be a rationale for why 11.2% is the right number, particularly since what we heard is the obstacle is not the absence of a program. The obstacle is CERN doesn't pay enough money and requires too much effort. And I don't see that addressed. And you've created a massive program that actually, I don't have any evidence that you've actually solved the problem. I'll stop there.
Thank you. Fred and Rosa, perhaps you can get through some of these issues offline. Leandra.
Thank you. I'll be quick. First, thank you. Thank you for the conceptualization and the time that staff put into this remind. I love it, by the way it's labeled. I do agree with Fred. There is some confusion here and I would like clarity. So whatever conversation or whatever you work through offline, if you could report back to the rest of us, for those of us that are not quite caught up, I would appreciate that. And then also to Fred's point around the money and the effort, that truly was what stood out to me in one of our last meetings. And, I know it was reiterated at our ICOC meeting a few weeks ago. And so I want to make sure that that doesn't get lost in all of this. And so I think that there's opportunities to continue the discussion. Also to Pat's point around the Community Advisory Committee, it's something that I raised at our last meeting, and I'm glad to hear it raised here again. I think there really needs to be space and room for it, especially considering the direct feedback that we've received, some of which Fred just relayed. So I do hope that there's going to be some efforts to do that. But again, thank you for this. I know you all are working this out. I'm glad we're having this meeting so you can hear directly from us what some of the kinks might be or better ways to make it clear, so that everybody, because if we're not clear, believe you me, others won't be either as this moves forward. So appreciate that. Thank you.
Thank you, Leandra. And thank you, Fred. Great feedback.
I just wanted to say that I think this is a really good start at the issue that we were trying to get to, is understanding why nothing had been done at all on neuropsych. And this is an effort to begin to cure that I think is actually pretty clear and outstanding. And my only question would be, is there any possibility of accelerating the timeline on this? Because it's a lot, but it's a long time, and neuropsych has been so under focused on that whatever we can do to move it forward, I think would be a great thing. But I think this is a good start, personally.
Thank you, Marv. Can I ask somebody a process question? We're having to wrap up here. Do we formally vote to send this on to the science subcommittee, or do we make a recommendation, or do we have any rule at all about what our our final act is here today?
Sure, Larry. This is Scott Beck at CERN. It's the pleasure of the committee, but in the normal course, the recommendation would be to forward to the science subcommittee with a recommendation to the committee and full board to adopt it.
Great. So we can make a voted recommendation. So can somebody make a motion, please?
I would move with after clarifications as to the slide, that this move forward to the scientific committee.
I second.
Thank you. Let's see, a roll call vote. Mary Ann?
Yes.
Just a second, Larry, sorry, if there's a board comment on the motion, we'll take that now. It looks like Steve's hand is raised.
Great.
And then
Steve.
we would have public comment after that.
Okay.
So Marv's motion actually opens the door to this question, because he didn't move that this presentation per se be forwarded to the scientific subcommittee, but with modifications. And I go back to something that, Fred was talking about, and Pat too, for that matter. Are we really ready for prime time with this? I don't want to go recommending something to the scientific subcommittee. And Leandra said the same thing. That's not quite fully baked. I mean, this is pretty important, what we are planning on doing. And it's not clear to me that we're at that point. And I'd rather take a little bit more time and beg the ICOC's indulgence ultimately in order to have something that we're all settled as, a good plan to move forward.
Fred?
What Steve said. If this motion does come to a vote, I will unfortunately have to vote no. It's not ready.
Maria Milan?
It may not answer all the questions, but I wanted to, I think, answer the question about this 11%. That 235 million that is on the salmon arrow, I believe, is a total expenditures for all discovery programs coming through the usual pillars for neuro, not neuropsych, all of neuro. And I believe that that got added to the proposed expenditures for these, these bigger programs, and that gave rise to the 648 million. And the balance of that would be what's available for other programs, including translational and clinical. So it may not make a difference, but I just wanted to point out my understanding of this 235 million. So the calculation.
I can explain, but yeah, I decided that we would clarify in a different
time
way.
to go back. We don't have time to go back and get into the granular detail. It's a good example of why we need another meeting to talk through all of this, because 235 million on neuro in the past cycle doesn't make sense either. So
No, I can explain, but I didn't think we had the time. So I think perhaps we have another meeting and I can make the clarifications that everybody's asking. This is easy, this was a big bite to bring into the task force, and I'm happy to clarify. It's easy. It's very easy, all these questions. And I appreciate them. That makes me realize what's not understandable.
Maria Bonneville?
I was just going to add, I think it's important at the next meeting on Moran team is to come back with answers to some of the things that were brought up outside of this specifically, but also, is it enough money? How did we come to those conclusions? What's the percent effort? How did we arrive at all of that? Because I think there has been in the past several meetings a call to research some of these issues that have been brought up on more than one occasion. So I think it'd be really helpful.
Steve?
Yes, I'm just going to basically reinforce something that Fred said during the course of his soliloquy. Have we socialized what we would like to do in terms of funding with any of the folks that have made presentations before, or other research institutions or academic institutions within the state. In other words, the question on the table is, are we willing to grant enough money to make a dent in this? Do we know that? I mean, so we've provided some amount of money in some time period. Is that really sufficient or not? I would like to know that people from outside of this group say, yes, you know, that's great, that's perfect, that's exactly what we need, or no, wait a minute, that's not quite enough, you know, we wouldn't take our time to apply for a grant. Because I don't want to run into that problem again. I want to, you know, make sure that what we do, we're going to be successful at doing.
The answer, Steve, is yes, we have. We have also been talking to the NIMH, which would be the funding agency at the federal level that has been granting awards like this. We've been doing a landscape analysis of what's funded out there. We've been talking to researchers. We have to be careful because this is not a concept even. So if we put too much sweetness in their mouth and then we don't give it to them, they will think that, but we have actually been talking to the people that spoke and others, and we've been going to meetings as well. And everybody's very excited and waiting for this to be out.
Great, thank you, Russell.
Pat.
Yeah, I was just going to comment that there's enough expertise on this committee to you know, there's not a formula. There's not going to be a formula that says we need exactly this amount of money to address the gaps that have been identified by those who have presented and what this task force has addressed. There's gaps in every area of brain diseases and disorders everywhere, right? So, you know, we've gotten input. There's certain pressure points that we know about, and the funding levels are going to be to some degree subjective. It's just the way it is. We can't predict that it's going to be 550 versus $750 million, and then if we go to 750, that'll be enough. Who knows what's going to be enough? Is there anyone on this committee who can tell me what the formula is? So I think we do have to have another meeting to give Rosa and the team a chance to make the edits and to suggestions, and maybe between now and that meeting, some offline conversations with some folks here about understanding the dollar amounts, which I think can be somewhat confusing. That's my recommendation, and then we have to get on with the boat. The expectation that we're going to come up with a formula that's going to tell us exactly how much money is going to be the right amount of money is just not going to happen. That's it.
Thank you, Pat. Scott, process question. Where do we go from here?
Well, the motion that has been made and seconded actually technically belongs to the whole of the task force now. Listening to the comment, I think maybe procedurally you could just ask if there is no objection that we table the motion until another meeting of the task force to see a refined proposal, and take up the motion at that time after the presentation at that meeting. And that we would schedule that meeting, of course, before the science subcommittee meeting.
And do we do that before or after we get public comment?
You could do that after you receive public comment, or for now, it's I think either is fine.
Let's see, is there anybody on the line for public comment? Because if there's somebody that wants to address these issues, that could be helpful.
There looks like there might be a couple, but if you are, and you can do *9 to be put in a queue if you'd like to make a comment. And there are none popping up, Larry.
No comment. So then I think we should do what Scott suggested if those who proposed the original motions are in agreement. Please let us know.
Fine with me.
Yes.
Okay, great. So next step is another task force meeting that will address the issues that have been raised about dollars, effort, socializing with the community, and some clarification of the timing and growth of Remind L and Remind X. Do I have that right?
Yes.
Good. Okay, Rosa, okay?
Fantastic. Yes,
Great, thank
we'll be
you.
there.
Okay. I think with
And
that,
thank you, Larry, and thank you everybody for the feedback. Very useful.
okay, so I think with that, we can adjourn and see you all soon with additional information.